Welcome to our video on amino acids. So in this video, we're going to look at amino acids, classifying them, and talk about the need to memorize those amino acids. And let's just take a look at one example of why we care. So myoglobin, shown here, is a peptide or a protein made up of 153 amino acids. And it turns out that the order and type of those amino acids give it its structure and function. When we look at this, you can see right here we've got our ribbon drawing. So this whole thing is our myoglobin protein. But right here we have this heme group. This is not part of the protein chain. This is separate and held into place. There's an iron atom in it. And this heme is what carries the oxygen. Well, it turns out that if you count from 93 positions from the N-terminal end, the amino end, you find a histidine. And that histidine's job is to hold this heme group in its spot. Okay, and there's another histidine, 64 from that end. Sounds like it should be far away, but because of the rotating and curving, they're really not that far away. And that second histidine is important because it keeps the iron from being oxidized so that it's available to bond the oxygen. So then we need to know why histidine. Like, why does the histidine carry that importance? Why does it do this job? And to know that, we need to know its structure. All right, so let's take a look at amino acids, kind of in a generic sense. So on the top left, right here, we have Fisher projection. So when we look at this, we've got to remember, wow, 1, 2, 3, 4 areas of high electron density. So I expect this to be a tetrahedral molecule. So I know it won't be flat, like shown. In the Fisher projection, we assume that the ones horizontally are coming out of the page towards the viewer, and the ones vertically are going back into the page away from the viewer. When I look at an amino acid, I can also draw it in kind of a stereochemical or stereo rendering so that you can see that 3D. The little wedge are coming out towards us, the dashed going backwards. All right, when I look at this, I've got an amino terminal, I've got a carboxylate terminal. Sometimes you'll see it NH2 and COOH, but the reality is at physiological pH, they're usually charged, so we often show them this way. This carbon right here in the middle is the alpha carbon. Okay, so between the two, attaching the carboxylic acid and the amino terminal. And then every one of our amino acids will have one hydrogen on that alpha carbon, and then something else. And here's where the different amino acids are going to differ. Okay, and so the one we're looking at here is alanine, but there are a bunch of them, and we're going to take a look at that. Important to know is that there are two isomers, right? The L and the D. They were R and S in organic chemistry, but in biochemistry, we tend to talk L and D. So what was S in organic is L, what was R in organic is D. And so these are mirror images, right? We got these stereoisomers, mirror images. And an interesting thing is that for most amino acids, only the L isomer turns out to be biologically active. We don't know exactly why it evolved this way, but it's true that the active sites of enzymes are asymmetric, and they're specific generally for creating the L isomer. If you create these chiral compounds in ordinary chemical reactions, not in vivo, then you would typically get a 50-50 mix. But in living systems, again, almost all L isomers. All right, onward. So amino acids will have a wide array of functional groups, as we mentioned, And you need to know the 20 amino acids that are found in proteins. And for each, you're going to need to know the structure, the name, the 3-letter abbreviation, and the 1-letter abbreviation. All of these are commonly used. And for us to be able to talk about proteins and structure, you're going to need to know these. We have to have that common vocabulary. We will also group these amino acids by the properties of this side chain. So they might be hydrophobic, polar, positively charged, negatively charged. This is something you really don't need to memorize because once you know the structure, you know enough chemistry to look at it and pick it out. So let's try it. So take a second and look at these amino acids and choose which classification you think they would go in. All right, when I look at these, I'm going to look at the side chains. So here was the one we looked at before with CH3. I would expect that to be nonpolar. And if I come down here, wow, a bunch more carbons and hydrogens, nonpolar, nonpolar, nonpolar. So we would consider these to be hydrophobic amino acids. Now, if you are thinking, but wait, what about this and this, right? Our amino and our carboxylic acid, certainly those are polar. And we're going to see that in our proteins, these things are going to be used to bond to each other. Kind of like Legos, they only go one way. The carboxylic acid end can bond with the amino terminal of the next. But as we classify them, we're classifying them just based on those side chains. Glycine, you'll see, is special because it just has those hydrogens. It's not actually chiral because we have the 2 hydrogens there. We're going to look at a couple of these specifically in just a second. This is just a little bit about what we just said, if they consist of only hydrogen and carbon, and then we're going to look at a couple of the other examples. Let's look at methionine. Sometimes people say, wow, sulfur carbon. Yes, it's a little bit polar, but not that polar, and that sulfur is buried between the 2 carbons, so this is largely still nonpolar. Proline is an interesting one because that side chain wraps around and forms a bond to that N terminal. This one's going to be important because this bond, this linkage, causes a kink in the chain. And so this can be an important amino acid structurally. And then if I come over here and look at tryptophan, you might look and be like, wow, there's a nitrogen there. But there's so much resonance in these that really that nitrogen is not reactive. It's kind of buried in that resonance, and so it doesn't act like an acid or a base, and this is largely still considered nonpolar. All right, let's go to the next ones. So take a moment, look at these, and think about how you would classify these. All right, as you look at this, I'm coming in here and I'm going to say, wow, I've got my hydroxyl group, oh, another one, right? Here I've got that sulfur again, but now it's got a sulfur hydrogen bond that's more polar. So I'm going to look at these and I'm going to say, you know what? These are polar amino acids. We're going to figure out that these hydroxyl groups, when you have the OH, those are also really reactive. And so those are very important in a lot of active sites. So as we look at this, over here, this is interesting because we have this amide functional group, but because this NH2 is bonded to that carbonyl, this amine group does not tend to act as a base. So it doesn't tend to do that, and hence we think of these as polar, but not necessarily charged. All right, let's keep going. These next ones are going to be the top question, a little more obvious. As I look at this, I'm going to say, hey, I've got this positive charge, positive charge. This is a little weird though, because it doesn't have a positive charge. So these all still get classified into this positively charged amino acids because they have the potential to have a positively charged side chain. As we're talking about these, and what we're going to find out in the next video is that it will depend on the pH if they will be charged or uncharged, but they have the potential. So we know from Gen Chem that that amino group, that nitrogen, tends to act as a base. It can accept a proton, and hence we have the potential for that positive charge. And because those things are charged, we are going to expect them to be hydrophilic, right? Water would be attracted to those. If we look specifically at the histidine then, so we have this side chain right here, and take a look. We know that that nitrogen can accept a proton or donate a proton, so let's look at that. Come back in here. So as we go this way, right, it's accepting a proton. I'm sorry, it's donating a proton. I said that totally backwards. So as it gives away a proton, now when we're over here, we have a neutral side chain. I actually missed part of the side chain, but you got it. As we go back the other way, right, now it's accepting a proton. We see that proton right here. And so we have that positively charged. So this goes back to the idea of weak acids and weak bases. If you remember, we talked about they were essentially a protonated and deprotonated version of the same molecule. And if you think back to weak acids and weak bases, we had our Henderson-Hasselbalch equation. That looks like this. So we know that each acid or base has a pKa value, and at that pKa value, we have equal amounts of the deprotonated and the protonated version, of this version and this version. So if we think of physiological pH, for now, let's just think of about 7. And if I told you that the pKa of histidine is about 6, I would look at this and you could think it through mathematically. Do you need to have more A minus or HA to make this work? And it's going to turn out that we need this whole value to be above 1, or right, to be above 1 so that we get a positive value so that we can make this equal to 7. And in fact, this ratio has to be 10 to 1 to make this work. So I know mathematically that I need to have more of the A minus. But you can also think about it more just kind of conceptually. If we have a 50-50 mix at the pKa, when pH is 6, we have a 50-50 mix. Above the pKa, usually higher numbers mean basic, right? So above the pKa, you're going to have more of the basic version, the deprotonated version. And so this is why up here we showed these two, because we're generally showing them at physiological pH. We showed these two as protonated, but this one, by pH 7, we've already bypassed its pKa, so we have more of the deprotonated version. It's going to turn out that histidine is important because that pKa value is so close to physiological pH that it can often act as an acid or a base. As we look at pKa's in the next video, you're going to see that often they are either really high or really low. There aren't a lot of them in the middle, like histidines. All right, and our last set of just 2 right here. And you see those negative charges. These are our negatively charged amino acids. So we have carboxylic acid side groups. Okay, so you could also call this aspartic acid. Okay, but we also often call it aspartate, right? Usually this is already charged and we call it that. A fun fact is that if we look at the other one, the glutamate over here on the right, and you think about monosodium glutamate, MSG, right, that is often used as a taste enhancer. So it turns out that the flavor of these two amino acids is really what gives us that flavor group. All right, one last thing for amino acids. Essential versus non-essential. This has nothing to do with importance. This has to do with, can your body make this? Can it synthesize this amino acid or not? For non-essential ones, your body, assuming it has the right starting materials, can synthesize that. For essential amino acids, you can think of it as essential that you consume those in your diet because your body cannot synthesize these amino acids. So we count on other organisms, plants, animals, whatever, to synthesize these, and then we consume them. All right, learning the amino acids is going to really, really help you. I would suggest flashcards. Make some good quality flashcards. Mnemonics help in remembering some of these. So take a look at that. There are apps you can do to quiz yourself. All right, and then we will look next at how pH combines with our knowledge about the amino acids to help us determine if a overall polypeptide might be positively or negatively charged.